This presentation is really following, because those of you who have paid attention or been really active into the field has seen that last week EHDN, the European Huntington's Disease Network, and the Huntington's Disease Association in the States had a webinar where Roche presented some of the same things that will be presented today, but today will be less scientific. Today will be more about the key findings, the lessons learned, what are the plans, and the rationale behind these plans. So I hope that makes sense to you all and that you will find it useful. And I'm really, really pleased to welcome Lauren Boak. She's a global development leader in Roche and has followed this program since, I think, the start, Lauren. At least we have known each other for several years. So please, the floor is yours.
Thank you, Astrid. I'm very pleased to be here with you all today. It's a much more personal view seeing all your faces. So thank you for joining. Yes, my name is Lauren Boak, and I have been working on Tominersen since Roche started actively running the program about 5 years ago, when we took it over from Ionis. So we've had a pleasure of meeting Astrid and many of you many times before. So great to see you again. And my colleague Peter, will introduce himself now as well.
Thanks, Lauren. I'm delighted to be here today to be able to share with everyone the update from Tominersen. My name is Peter McColgan. I'm a HD clinician, and I've been looking after HD families for nearly 10 years. I was previously at University College London, and I've been working on Tominersen going back as far as 2015. I was one of the original investigators on the phase 1 study. So I'm delighted to be able to be here and share this update with everyone.
Thanks, Peter. So yes, as Astrid outlined, we've shared an update from the program last week, and we went into a lot of detail at the EHDN and the HDSA events or presentations, and so we won't have as much detail today, but we're really looking forward to having ample time for questions and answers at the end. So please get ready for those, get those questions ready for us. Just to start, if you just go to the first slide, please, Peter. I just want to start with a really important message, and that is just the gratitude and thanks to each and every person in the HD community. In particular, patients, participants of the studies and families. What incredible trust you put in researchers and programs like the Tominersen one and all of the others in the field. So just want to start with that really important thank you and gratitude before getting started. Next slide. Thanks, Peter. And just this is a disclosure we need to do at the beginning because what we share is not intended as specific medical advice. Tominersen is still a research or investigational medicine, and that means it's not approved for use. And if you in particular have some questions about your own care or potential treatments, please contact your healthcare provider. Next slide. Okay, so yes, as mentioned, we shared news last week about the plans for the Tominersen program to continue and start a new phase 2 trial. And this was publicly announced and quickly followed with information sharing with the investigators and study sites, so that this information about the analyses and the next steps could be shared very quickly thereafter with participants and families directly. So the doctors and healthcare teams are really the best ones in the position to answer the questions that participants and families have. And just want to sort of start with this because sometimes it's a bit confusing as to why something like this that's so personal to everyone in the HD community is shared publicly. But this is how we approached it, and I hope that helps with that. Now, just next slide, thank you. So today it's a truncated presentation. We're not going to go into the all of the details. As Astrid said, there's a lot of information, and it's taken months and months for the Roche team to really sort of analyse and try to make first steps to understand. So being able to understand it, just even the scientist ones through, it's very difficult. So today we really hope to just give you key information and insights to help you understand where things stand. I'll start with a very brief history, where we stand with the actual ongoing studies, and then pass to Peter, and he will share some top-line findings, not all of them last week, and if you want details, go to the presentations. But I just want to mention one thing, and I think this has been a question that we've had, and it's understandably a really good question. Why haven't we been sharing everything right now? And I mean, there's just so much data, and we have to, take it step by step in our looking at this data, understanding it, but also how we share it. And so, but rest assured, we'll be sharing all of the data and progressively as we can over the course of this year. And then Peter will share also what this means for our next steps in planning for Tominersen. Next slide, please. Not next step. But this is a summary of the program to date for Tominersen. So we started with the phase 1/2A study, and then the patients from this study went into an open label extension study. These studies have been complete, the OLE has been complete for a while. We also had a natural history study, which was a study that didn't involve Tominersen treatment, but was really evaluating key scientific questions about the changes on certain biomarkers and clinical endpoints over time. The study has finished, and the final analysis is ongoing. Then we've got the Generation HD1 phase 3 study that unfortunately dosing needed to stop in March last year, but this study is still importantly ongoing. Safety and follow-up is still continuing for participants in this study. This is also the case for the open label extension GenExtend study. And so this is ongoing to following patients for safety and outcomes. The last study is GenPeak. It's the smallest of our studies, but still a very important study. This is a PK/PD study that was done just to really look more closely about what happens to Tominersen in the body, and then what effects Tominersen has in the body. This study, first part of this study has been completed with the 12 patients recruited for this study, and we've decided, because of the really important information already collected, or the data from this study, that this can stop now. We don't need to conduct part 2. So this study will move forward through to closure and reporting now too. Next slide, please. This is just a slide just to demonstrate how many participants were in the study at the time, the study Generation HD1 and GenExtend, at the time dosing needed to stop in March last year. So there was 791 patients recruited in this study, and nearly all of the participants were still in the study. So this is just, as well as also during the period of follow-up, over 80%. And this is the same for GenExtend as well. And this is really, these are really impressive numbers of retention in the study, despite the fact that we need to dosing, and I think there might be a bit of feedback. And this is just a real testament to the commitment of all investigators, sites and participants, that this staying in the study even after dosing is really important to help us understand. And so we really appreciate that. But this also, appreciation, a bit of feedback again, sorry. Appreciation extends to everybody that's been in the study, as I mentioned before, in studies. Next slide, please. Okay, so this is just a slide to the update on the status of the Generation HD1 and GenExtend studies. We're pleased to share that the Independent Data Monitoring Committee, which is the oversight committee for Generation HD1, have reviewed data after dosing in the Generation HD1, and their assessment is that we don't actually need to have any additional follow-up outside of the actual planned follow-up in Generation HD1. So that means we can move to close the study, and we're also going to do the same for GenExtend. So the last patients visits will happen in the by the time in the next couple of months through March and April. These studies then will start. The closure activities that happen at study sites at the end will happen. That's where we need to look at just the quality of the data and make sure all paperwork is done. And then the final study closure checks that need to happen. So what this means, in particular important for the Generation HD1 participants, is that all participants will be able to be unblinded by the end of June at the latest. And that means that they will be told whether they received placebo or Tominersen during Generation HD1. We're very appreciative of the patience of everybody, of participants, knowing this information. It's probably been important for everyone, but it's been also equally important, well, it has been important for us for the study to really collect the data in this blinded fashion up till now. So this, please, we're able to give it a month now when this is going to happen. There's just one thing that there is if in case participants are looking to see if they're eligible to participate in another study, another Huntington study, then they can talk to the investigator about this, because they may be able to receive this information before June. So this is a really important period of time for participants in Generation HD1 and Gen Extend. Lots of, it's been a long journey work on these studies, and we really appreciate this. And knowing now, it's probably the question, what happens now to participants? And obviously the final visits, if they haven't happened already, are important. And at the moment, we are still discussing what, in the context of the new study that Peter will outline, whether or not participants that have previously been part of these studies could be considered for inclusion. But at this point in time, we're very early days in the planning, and so as soon as we know any more, we will share this information with your site and with your investigators. So thanks again for your patience and understanding that we just haven't got all the answers just yet. Okay, so that's the status of the studies and next steps. So my last couple of slides are just a reminder of what the Generation HD1 study is. So a bit of a focus on the Generation HD1 study and what were the results. Well, what happened when the IDN, the Independent Data Monitoring Committee, recommended to stop dosing in March. Next slide, please. March last year. So this was a Generation HD1 study, was a large phase 3 study. It was conducted in 18 countries worldwide, over 100 sites. So it was a very geographically spread study. This study recruited 791 participants. The key inclusion criteria included being having adult onset HD and an age between 25 to 65, as well as a CAG age product or CAP score of greater than 400. Peter will go through a little bit more about that when he comes to his section. It was a 3-arm study. It was a placebo-controlled study. So that meant that we had one arm, participants were randomized to either receive placebo or no drug, or one of 2 different dosing regimens of Tominersen, either 120 milligram every 8 weeks or 120 milligrams every 16 weeks. The original plan was for this study and the treatment period to last for 101 weeks or 25 months. And unfortunately, due to the Independent Data Monitoring Committee review of data up to the point in March 2021, so last year, we needed to stop dosing, and the studies didn't stop though. The study didn't stop. The studies are still continuing through to March, April this year, as I mentioned, and this is to evaluate clinical and safety outcomes. Now, the reason and the rationale behind this need and the disappointing news that we all shared the disappointment of last year is on the next slide. And that is due to, based on a review of the data available, the IDMC concluded that based on an overall benefit risk assessment, that dosing needed to stop in both dosing arms, or all study arms. And what we mean by that is that the good effects or potential good effects of Tominersen there wasn't a good balance between good versus bad, or putatively bad effects. And so in the 120 milligram every 8 weeks, and this is the more frequent dosing regimen, the data showed that Tominersen was unfavorable compared to placebo. And in the 120 milligram every 16 weeks, whilst the overall benefit risk was not clear, the safety profile actually appeared comparable to placebo. But when looking at whether there was any benefit, there was not an apparent benefit versus placebo. So on balance, given that there was no positive benefit risk, this dosing arm was also stopped. Okay, so that's a bit of a recap of where we stand with the programs. And with that, I will be passing to Peter to go through what we've learned since then. Well, some parts of what we've learned since then.
Thanks so much, Lauren. So I'm going to take you through some of the post hoc analyses, some of the data. Before I do that, I want to explain what a post hoc analysis is. So when we design a clinical trial, so if we consider the Generation HD1 study, we design it to test a specific hypothesis. In the context of Generation HD1, the hypothesis was, could Tominersen have a beneficial treatment effect on patients with manifest Huntington's disease? And so the study was conducted, and as Lauren has reminded us, unfortunately, due to the IDMC's recommendation, dosing was stopped back in March. When dosing was stopped, we analysed the data and we presented some of that data at the CHDI conference in April. And that data showed, as we've been reminded, that patients taking Tominersen every 8 weeks had an unfavourable outcome compared to placebo. That's the group that weren't taking Tominersen. Whereas patients taking Tominersen every 16 weeks, the outcome was similar to placebo, suggesting there was no benefit. Now, when we had those results, the next step for us was to try to understand, is there a subgroup of patients that may have benefited from Tominersen? And in this context, we come up with a new hypothesis. And that new hypothesis may be a particular subgroup. Could that subgroup benefit? It's important to say that this is when we design a post hoc analysis. And it's called a post hoc analysis because the original study was not designed to test this new hypothesis. Therefore, any of the results that we have from a post hoc analysis are not definitive. The study's not designed to test that new hypothesis. And so while the data that we share today is encouraging, I want to reiterate that the results are not definitive and they would require further exploration in a new phase 2 study. And that's exactly why we are proposing a new phase 2 study based on the post hoc analysis, because the results are not definitive currently. So as I mentioned, the first thing that the first task we had was to consider, could there be a subgroup of patients that may have benefited from Tominersen? And in addition to the clinical outcome data, does the other biological data, for example, CSF neurofilament light and CSF mutant Huntington, does that support the subgroup that we're looking at? And I'll come on to explain more about that. So we began our post hoc analysis with two core hypotheses. Could outcomes on Tominersen be affected by patient population? And by that, we specifically mean the stage of Huntington's disease patients have, or the disease burden that patients have. Disease burden can be measured by your age times your CAG repeat length. And I'll come on to explain a little bit more about that as well. So that was one part of the hypothesis. The other part was, could dose affect the outcomes on Tominersen? For example, is there a difference between patients receiving Tominersen every 8 weeks and every 16 weeks? And also, is there a difference based on the amount of Tominersen in the body after we inject the drug? And that's an analysis, an analysis called the exposure analysis. And we'll share more about that very, very shortly at a future scientific conference. Now, we did the analyses in 2 steps. We first did a univariate analysis. This is when you look at a single variable. So for example, if part of my hypothesis is, do older patients have different outcomes to younger patients, then we can look at all the patients enrolled in the study and split the group of patients in half based on their age. So one group would be high age and the other group would be low age based on a split down the middle. So this would be a univariate analysis. A multivariate analysis is the next step, and that's when we look to see how 2 variables interact and such that they may affect outcomes on Tominersen. For example, instead of looking at age, we can look at age and CAG repeat length and see if those have an impact on the outcomes. When we did these univariate and multivariate analyses, we found 2 results based on treatment. So for patients taking Tominersen every 16 weeks, there was a potential benefit in a subgroup of patients who were younger with lower disease burden. However, importantly, while that result was encouraging, it requires confirmation in a new study that's specifically designed to test this new hypothesis about younger patients and lower disease burden. On the other hand, in patients taking Tominersen every 8 weeks, we found that outcomes were unfavorable regardless of the subgroup that the patients were in. I just want to take a minute and explain about disease burden. So disease burden, as I said, can be measured by your age times your CAG repeat length. And this is known as the CAP score, or CAG age product score. It's important to say that this is a research tool. And while this relates to the disease burden or the extent of disease in Huntington's, there are many, many other things that impact the extent of disease. And so this needs to be interpreted with caution. And I'll come on now to explain about the subgroups. I want to say first off that the subgroups that we are looking at here are based solely on the data that we have in the Generation HD1 study. You'll see here that when we looked at the patients in the study and divided them by age, the middle age was 48. When we divided based on the CAP score, the disease burden score, the middle cutoff was 500. This does not mean that ages below and above 48 are representative of what a high and low age would be in the Huntington's community. And it's the same for CAP score. This is just based on the data that we have. The other thing I want to say is that this is the subgroup that we found based on the treatment regimens and doses in the Generation HD1 study. It does not mean that lowering Huntington and other Huntington lowering approaches cannot provide benefit in patients who are older or patients who have higher disease burden. I appreciate that the news about the lower age and the lower disease burden may be upsetting for some people, but I want to reiterate again, just because we haven't found any potential benefit in this subgroup doesn't mean that patients who are older with higher CAP scores can't benefit from these approaches. With that said, I want to explain the subgroups that I'm going to talk about in the data portion of the presentation. So when we split by age and CAP and the patients enrolled in the study, we were left with 4 subgroups. Group 1, low age, low CAP. Group 2, low age, high CAP. Group 3, high age, low CAP. And Group 4, high age, high CAP. And these are the 4 subgroups that I'll show some of the clinical endpoint data on. I'm going to explain the different measures that we looked at in this analysis. And if you want to see more details in terms of the data of these measures, you can see that in the HDSA presentation or the EHDN presentation that we did last week. I'll focus on the clinical outcome measures in this presentation, but I'll explain what the others are as well. So the clinical outcome measures are different tests that we use in Huntington's disease to see what stage people are at and how they're progressing. These tests come under an umbrella called the Unified Huntington's Disease Rating Scale. And there are 5 different tests that we look at, and I'll explain these in detail as well. When we sample the cerebral spinal fluid, anyone who was involved in the trial knows that we take some fluid out and then we inject the drug. And that fluid we take out, we look for 2 main different things. One is neurofilament light, and that is a protein in the cerebral spinal fluid that represents any cell loss in the brain. The other thing we look at is mutant huntingtin, and mutant huntingtin is the protein that causes all the problems in Huntington's disease, and we can measure that in the cerebral spinal fluid. The other things that we look at in the study are brain images using an MRI scanner, and so we can look at ventricular volume, and that is a measure of how much space there is in the brain. And then finally, we look at safety. And I'll summarize what all these measures show near the end of the talk, but I'll focus on clinical outcomes for this presentation. So for the clinical outcomes, I'm going to focus on the Composite Unified Huntington's Disease Rating Scale. I'll show the group level data, and then I'll go into the subgroups, and I'll talk specifically about the low age, low cap subgroup where we see potential benefit based on the post hoc analysis. I want to explain first what the Composite Unified Huntington's Disease Rating Scale is. So this is a composite, means that we look at 4 different measures and we combine them together. The 4 different measures are total functional capacity. This is when your physician will ask you questions about your ability to work, your ability to look after your finances, your ability to do chores at home, and the amount of care you need at home. The total motor score is a test that the physician does when they examine you, and they'll look for the motor signs that you have that are specific to Huntington's disease, like chorea or your ability to walk and things like that. The other 2 tests are cognitive tests. They measure your ability to think, and specifically the speed of processing. So that is the symbol digit modalities test, and this is where you match symbols to numbers, and also strip word reading, and that's your ability to read the names of colors and how fast you can do this. And we take all these scores together and we come out with the composite UHDRS. I'm showing you the group level data first. So this is all the patients in the study. If we look on the y-axis, we can see change from baseline in the composite UHDRS. If we look on the x-axis, we can see the time points. So week 5, 21, 37, 53, and up to week 69. The different bars here are the different groups. So gray is placebo. This is a group of patients that didn't have treatment. Orange is patients having treatments every 16 weeks, and blue is patients having treatment every 8 weeks. If we focus on week 69, as this is the last time point that we've analyzed, we can see that patients taking treatment every 16 weeks are slightly below placebo, but the bars here, the confidence intervals overlap, so we can say that this is comparable to placebo. However, for patients taking treatment every 8 weeks, it is much lower than placebo in the unfavorable direction. So going up the ways would be an improved outcome, therefore going down the ways is an unfavorable outcome. So the take-home message for this is that Q16 was similar to placebo. Treatment every 16 weeks was similar to placebo. Treatment every 8 weeks is unfavorable compared to placebo. And just to say that this is the full data set. We presented the partial data set back in April, and now with the full data set, the pattern is exactly the same. So moving on to the subgroups, and starting with the low age, low cap subgroup. Now, this is a subgroup where we see potential benefit, but as I've said before, this is a post hoc analysis. The results are not definitive. The results are not statistically significant, and so they would require further exploration and confirmation in a new phase 2 study, specifically designed to test this group of patients. If we look at week 69 for the low age, low cap group, we can see that the circle in orange, that's treatment every 16 weeks, is above the circle in grey, that's placebo, patients that never had treatment. And so there's a suggestion that there may be a potential benefit. But as I've said before, because the bars overlap, this means that this is not significant. And while there's a suggestion and while this is encouraging, it would need to be tested in a study specifically designed in this patient group. We can see that in patients having treatment every 8 weeks, the outcome is in the unfavorable direction. And if we look at the other subgroups, low age, high cap, we can see for treatment every 8 weeks, the outcome is consistently in the unfavorable direction. If we look at the high age, low cap group, again, the outcome for treatment every 16 weeks is again in the unfavorable direction. And if we look at the high age, high cap group, the outcome every 8 weeks is again in the unfavorable direction. So the key take-home message from the subgroup analysis is that there is a suggestion of potential benefit in patients receiving treatment every 16 weeks in the low age, low cap subgroup. However, it's not statistically significant and it would need confirmation in a study designed to test this specific subgroup. However, for treatment every 8 weeks, regardless of a patient's subgroup, treatment outcomes are in the unfavorable direction. So that was the composite unified Huntington's disease rating scale. The other clinical endpoints show very, very similar patterns, and I'd again direct you to EHDN or the HDSA presentation for more details on that. But the takeaway message is that in the patients receiving treatment every 16 weeks, in the low age, low cap subgroup, there were favorable point estimates compared to placebo. And we see this on all UHDRS clinical endpoints. I've just shown one of them here, but we see this on all of them. And this is supported by safety and biomarker data. Let me explain a little bit about the biomarker data first. So for the CSF neurofilament light data, this is the measure of brain cell loss in the cerebral spinal fluid. For patients receiving treatment every 8 weeks, there was an increase at all time points, but the biggest increase was seen at week 21, and then levels dropped then back towards baseline at week 69. So an increase at the start and then levels coming back towards baseline by week 69, suggesting improvement, but potential a sign of damage at the start of week 21. For patients receiving treatment every 16 weeks, there was a small increase at week 21, but levels were in line with baseline at weeks 37 and 69, suggesting that there was less or no damage, at least at week 69. For CSF mutant Huntington, this is the protein that causes all the problems in Huntington's disease. We saw reduction in CSF mutant Huntington. The greatest reduction was seen in treatment every 8 weeks, followed by treatment every 16 weeks. And in terms of brain imaging or MRI scans, we saw increases in brain ventricles. These are spaces in the brain. The greatest increases were seen with treatment every 8 weeks, with less increases seen with treatment every 16 weeks. It's difficult to understand why we see this increases. It may be related to the injection of the drug into the CSF, or there may be other reasons, and we are currently looking into this. And then just to finish off, to say about safety. So in our low age, low cap group in patients receiving Tominersen every 16 weeks, the number of adverse events were similar in the treatment group compared to the placebo group. There were less serious adverse events in the treatment group compared to the placebo group. So the safety data is encouraging. So I just want to summarize what I've discussed. So it's important to point out that the Generation HD1 study did not meet its primary objective. So as I mentioned before, in treatment every 8 weeks, there were unfavorable outcomes compared to placebo. In treatment every 16 weeks, outcomes were similar to placebo, suggesting no apparent benefit. However, in our hypothesis-driven post-hoc exploratory analysis, in patients receiving treatment every 16 weeks in this low-age, low-cap subgroup, there were favorable point estimates compared to placebo on all UHDRS clinical endpoints. And as I've just mentioned in the slide before, that was supported by safety and biomarker data. Importantly, and this is really the take-home message from this, these findings suggest that low exposure Tominersen may benefit younger adult patients with lower disease burden. However, investigation and confirmation in a prospectively designed randomized control study is required. So what are the next steps for Tominersen? We are in the early stages of designing a new phase 2 trial to explore safety and efficacy of different doses of Tominersen in a younger patient population with less disease burden. The safety data of low exposure Tominersen, together with a potential benefit in the specific patient subgroup that we've identified, support the continuation of the Tominersen development program with a new phase 2 clinical trial. Details about the study design will be announced once finalized, including eligibility criteria, planned start date, and study sites. And just to reiterate what Lauren said about the current study, so for Generation HD1 and Gen Extend, last patient visits will take place in March and April of 2022. For Gen Peak, all patients have completed part 1 of the study, and part 2 will not be conducted. With that, I would like to say a massive thank you to all the patients, their families, site investigators, and everyone who has been so dedicated and committed to all the Tominersen studies. It's because of your commitment and dedication that we can share these results with you today that give us confidence there may be a path forward for Tominersen. So I want to say a big thank you.
And thank you, Peter and Lauren, for presenting this exactly as we had agreed to, just take the overview and the most important ones based on what you are intending to do next. So thank you so much for that. Maybe you can stop share screen. I think that's so we can see each other better in a way. And I will welcome Professor Anne Rosser also to this kind of mini panel, I would say. Anne Rosser is a professor at the hospital in Cardiff, and she's also the current chair of the European Huntington's Disease Network. So I'm really grateful that you are able to be here together with us, and as a kind of a clinician independent from the company itself, and give your input and what are your I guess main take-home messages here from what you hear from Roche.
Thank you, Astrid. So, and thanks for that presentation, Peter and Lauren. So I have got a few thoughts that I'd like to just share with you. And I should start by saying, as Astrid said, I'm an HD clinician. I was involved in I was a site PI for the Roche study. So I've got a little bit of insight to the Roche study, but that was the extent of my involvement with this particular study. So I just have a couple of thoughts. Now, I want to just first, put aside for a moment the analysis we've just heard, because I think it's worth, first of all, reminding ourselves how much will be learned from this trial. Because we've learned an awful lot. We've learned that we can successfully undertake large multi-center trials in Huntington's, and we know that now. We didn't know that before, and there was a lot of scepticism before this trial took place about whether these sorts of large-scale trials would be possible in Huntington's. So we've learned that. I think there will be a lot of lessons learned from this about the best way to move from early preliminary studies to large phase 3 studies. And this sort of learning is easy in hindsight, but it's not so obvious at the time. And it'll be important for lots of other trials, you know, trials of all sorts of other therapeutics. And I think we've learned something about the previously unrecognized adverse effects of intrathecal ASOs, and there will be a wealth of data that I'm sure Peter and Lauren could expand on this, but clinical data, spinal fluid, scans, which will teach us a lot more over the next year or so. So secondly, just turning to what we've heard today, and these are just my impressions. I mean, personally, I'm encouraged that it's worth pursuing Tominersen further from this analysis. You know, it's been made very clear that these aren't significant results. The point of this analysis is to try and understand whether whether no further work happens in Tominersen, or whether there is enough here to suggest that it would be premature to stop exploring it. And of course, with in exploring any therapeutic agent, it's important to look at the data very carefully and to balance assessment of risk with the risk of throwing the baby out with the bathwater. So then the question is, how should this be done? And that's much more difficult. And as you've heard, that Roche are going to take some time to work out how to do the next study. I think that, like I say, I think there's enough encouragement here to move forward to look at doses. One interpretation of the data is that the dose used in Gen HD1 were too high, so it seems entirely logical to me to revisit this in a careful and systematic way. There are other explanations for the adverse findings in the Generation HD1 study, and to what extent these can be addressed by analysing the existing data more, or by new laboratory studies or new clinical studies, I think is something that's an open question at the moment that I'm sure the Roche team are working very hard on. And so there are questions that will need to be worked out as we all develop more understanding and have further discussion. And I should say that I very much appreciate the willingness of Roche to share these findings at this stage, and also it's great to see how keen so many clinicians and researchers and families are to engage with this process and to try and understand the data and how we can best take this forward.
Thank you, Anne. Interesting. I'm thinking, because there has been some, questions about, now we have gone through a phase 3 more or less to the end, and then a new phase 2. Has that been done previously in other drug development processes, programs, as you know? Going back, starting not from scratch, but going back one step and then explore new phases again?
Yes, I don't think HD will be the first disease to do this. Lauren and Peter, I don't know whether you have specific examples you can share.
I can just share that drug development is not so easy, and particularly in neuroscience. And I think there's probably more examples than not. I mean, I think we are really very much still in the learning phase, and I think Anne, you mentioned it nicely before, like what we can learn from this about moving from early stage trials into the phase 3 trials is important. And we had the decision for Tominersen to move directly into phase 3 was based on information and data at the time, and it made sense based on what we knew. But what we've learned now is that perhaps skipping what can be a more typical stage 2 or phase 2 with looking at different doses was needed. So it's always just a balance on what information is available at the time as to whether to move into a phase 3, and the confidence was there at the time. But I think drug development and in neuroscience and in HD, it's unfortunately not as easy and straightforward as we all would hope. And we're just here to learn and learn as much as we can from this program and others.
And I should, thanks Lauren, that's really helpful. I should say there are other examples in HD where people have done earlier stage trials, had to return to the drawing board, and there are quite a few of those. And you know, you may have heard presentations of the Vaccinex trial, where you know the trial again has some positive indications, but the overall trial was negative. And you know, you have to look at the data and try and understand whether it's worth going back, and they're going through a similar sort of process. So that the process in general isn't an unusual one.
Which is interesting and important to know, of course. I'm thinking, Lauren and Peter, because I remember when you started or you launched the phase 3 trial, you had dosing every 4 weeks. And then quite quickly you decided to halt, and then you reintroduced the trial with every 8 weeks and 16 weeks. So already very early in the program, you noticed that, oh, maybe not so often as we first assumed. Can you just remind us a little bit about the rationale behind that decision, and that may be also valuable now in terms of, okay, maybe not 8, even 16 is sufficient in a way.
I can do that. Peter was still at UCL when that happened on the other side. So yeah, when we started the phase 3, we started with 120 milligrams. We wanted to go to the highest safe dose, and we also wanted to test 2 different frequencies, so every 4 weeks and every 8 weeks. Again, because we still didn't know, still don't know what would be appropriate. And at the time, once after we started, we had some more data come in from the open label extension study, which was from the original phase 1/2A study. And we saw that the every 4 weeks regimen was not being well tolerated, or wasn't safe. And the open label extension study is not a phase 3 study with a placebo control, so it wasn't something that we can determine really about sort of benefit, but we can definitely evaluate safety. And so based on that, we made the decision together with our steering committee to shift the dosing and to a less frequent regimen. And that was what led to that quite quick change, and it was important at the time, and as we're learning, even more important than ever.
But just to make it clear, the plans for the new phase 2 will also imply that people will be injected in the spinal fluid. That's the way to dose people with the medicine, isn't it?
Yeah. So we're in the early stages of designing the new phase 2, but the delivery mechanism would be the same. It would be an intrathecal injection, so via the CSF. That would stay the same. And in terms of, from what we've learned from the current post hoc analysis, that will guide us in terms of how we design the new study. And we completely appreciate and understand that the idea of that we see benefit in younger patients and patients with lower CAP scores may be very disappointing to patients who are older and patients who have higher CAP scores. And so I just want to acknowledge that. And it doesn't mean that the Tominersen or certainly all the other Huntington lowering approaches won't work in patients who are older, but the potential benefit that we've seen is in this subgroup. And so the next step for us is to look at Tominersen in this subgroup.
Which is really, I mean, to me, it kind of makes logical sense that it's a smart thing to treat people early. But I want to get back a little bit to what is this disease burden concept. You said it's about age, And then it's the CAG, so really the mutation that kind of repeats you have in your gene.
Yeah.
So it hasn't to do with, if I understand you correctly, it hasn't to do with how far into the disease you have come. I mean, in terms of having symptoms. Or
so these things, so these concepts in Huntington's disease research are all very much related. So if we think about it, the way I think about it is if you have a CAG repeat in the range that causes Huntington's, the older someone gets means that the longer they have the protein, and so the more effects of the protein. And so there's the CAP score, or the CAG repeat length, and age are related to the stage someone is, so the measures are all very much related. But in our analysis and in what we've identified is that particularly the CAP score in this lower age group is where we see potential benefits. So that's where our focus is currently. But to answer your question, CAP and stage and disease burden, they're all related to each other.
Yeah, because this is of course of great interest to the community, who will be or who can see themselves as potentially eligible or a target for a new trial. Mm-hmm. So I don't know. There are some comments in the chat. It's a lot about the data sharing. You did touch upon that, Lauren, that you will, or Peter, you too, that you are going to continue to share more data, because you have really in this program collected an enormous amount of data. The trial participants has been wearing, digital variables and really done a lot of things to report back on how they are doing. So maybe you can just say a little bit more on that. Yeah.
We are committed to sharing data, and it's we appreciate, particularly with the digital, that patients spent a lot of time, and it was in some ways it was a lot for them to do during the study, and I understand, and we understand that the release of the digital data is highly anticipated, and so that's the next portion of the data that we want to share. Encompasses things like the exposure analysis that I mentioned during the talk, but also the digital, and also what happens when patients stop Tominersen, where we have the dose halting and that. So we want to share that as soon as we possibly can, and we are working extremely hard at the minute to get that data together to be able to share with community as soon as possible.
Thank you. Is it possible for you to say something about the timelines? How much time would you need to plan a new phase 2 before potentially you're able to start? Can you indicate anything?
Yeah, we're not in a position yet to sort of give timelines. We don't want to put a timeline in front of you and then not meet it. And so we're really all I can share is that we're working as fast as we can. There's a number of steps, like we need to finalize the data, the design of this, what the study will look like, what type of inclusion, exclusion criteria we need, the doses, how long it will be. We also then need to talk with some regulators, and that needs to be approved. Well, we need to have approval with all of the Roche people that review, and our steering committee will be looking at this, and then the regulators need to say approve it to start. And then once that happens, and there's a period of study setup and finding the sites, etc. So it's got a bit of a process still. So we're very early days, but we're obviously, this is a top priority, and we're working as fast as we can. And as soon as we can share more concrete dates, as we know that's obviously of interest, we will.
Okay, thank you. Yeah, I realize that develop drugs and doing clinical trials is an experiment. So you don't know, and you need to test, and then you don't give up. which is encouraging for me, and I think for the community in general. It's encouraging because we did fear when we got the setbacks from both Roche and from Wave last year that, oh my gosh, is Huntingtin lowering not the right pathway to go down, and this is encouraging, really, that there may be some potential here still that needs to be explored. So Anne, I don't know if you have any final comments or thoughts you will share with us.
No, I think it's going to be a very interesting time, and I think Lauren and Peter were very keen, I think, as a community to continue engaging in this discussion, because I think it's incredibly valuable, not just to help move the science forward, but I think it's really valuable for keeping the families in the loop and understanding better what's going on, so they're better placed then to make decisions for themselves.
That's really something I can agree to. It's really valuable for us, and that you come here and spend your time and share and try to explain to us non-scientists about what your learnings are and what your plans are is highly appreciated, and the commitment, of course. So we are at the hour that we had set for this. I want to thank the presenters and Anne for being here and sharing their expertise and knowledge with us, and for you all for being here and learning together with us. That's important, that we educate ourselves and can take informed decisions about whether to join into a trial if possible or leave it. But I would definitely say, like Lauren and Peter did at the beginning of this presentation, a deep felt thank you to the trial participants, because it's because of you that we move things forward. Without us, without you participating and contributing and dedicating time and do everything it takes to take part, we wouldn't learn these lessons. And one day we will get closer and have more efficient treatment for Huntington's disease. So as Alba says, stronger together. We are here, we're a strong team, and we are here to back each other up for all good efforts, and we will get there. So with those words, I will say thank you so much, everybody, for being here tonight. And you can take out your camera and wave to us. It's nice to see you. Thank you so much.
Thank you.
And good night.
Thanks, everyone. Thanks.